Welcome to those online. We are going to get started with Professor Pablo Fernandez Peñas, who is both at the University of Sydney and Westmead, and he's going to talk about the multi-omics for skin cancer. The floor is yours.
Thank you. Thank you. Thank you for inviting me. And I hope that this is going to work well and we're not going to have issues. This is a very interesting topic, and I'm going to first show my conflicts of interest. Yes, I have a relationship with multiple pharmaceutical companies. Yes, work for them, being an advisory committees, giving lectures, try to get science and business together, but from the medical point of view. With this project that I'm going to talk, I realized that I know nothing. So I hope that you can help me with some of the other things. And also, I am involved in many clinical trials, and you've got the list of different companies and research grants. So dermatology is a very complex specialty just because of this. For those that you're from my background, probably you know what is in common in all this, but many people will look at this and will say, oh, there's people in on this, or there's some colors, or there's something, but it's too difficult to understand what is in common. You need to know a lot. And for those of you that you haven't found yet, all of them, they're Spanish painters. Step one. And that's what a dermatologist does. A dermatologist look at the skin and reads different things and get one diagnosis, but also we need to find the one that doesn't match. So we look at this is a skin. And you've got two things. When I show this to my students, so I show this to nurses and I say, what's the patient that you need to follow because he's going to develop a skin cancer? They look at the thing and they say 50 one side, 50 the other. And really, a dermatologist, that I know that there's one in the room, will say, no, there's only one patient that is a high risk. That is the patient that you have on your right. Okay, so that's the one. But even though the patient on the left may have one skin cancer, and in particular, a skin cancer that is a killer called melanoma. Melanoma is the fourth most common diagnosed cancer in Australia. We're the world leaders. Well, that's very bad. But sometimes New Zealand takes the lead, so that's good, because then sometimes they're the world number one. Kills a lot of people in this country, still increasing the incidence, you see on the graph on the right. And it progressed just from a simple pigmented lesion. So you could see it. Many people could see it. In the past, we used to have pamphlets that say, have you seen this killer? And it was photos of melanoma to teach people what they need to look. And we know that the sooner that we detect this skin cancer, the better. The problem is that even for doctors, it's very difficult. You've got a photo on the right of this man with multiple scars, multiple moles. We know that doctors, when they need to remove things, they need to remove between 3 and 287 to find the melanoma. Well, it's not that bad. I don't know who published that one. That was in a recent review. The average or the mean number for most clinicians will be around 15. So we remove 15 things to find one that really needed to be removed. If you go to a dermatologist, you're way better. It's only 7, between 7 and 8. Well, if you go to our high-risk melanoma clinic at Westmead Hospital, it's only 3. We're way better, but still there's room for improvement. We're still doing surgery that doesn't need to be done. ACRF ASMID acronym. This is what the project that I'm involved now. So a group of universities, the University of Queensland, the University of Sydney, and Monash University, we got together and we applied to the Australian Cancer Research Foundation for the creation of this Center of Excellence in Melanoma Imaging and Diagnosis. And this is based in this device. Imaging. So you've got now in front of you a 3D photography system that when you stand in the middle, it depends how much skin you want to show us. If you want to show all the skin, that's okay. If you want to show just partial skin, will be that partial skin. We take one photo and the system can build a 3D avatar of you. So that's what we're going to do. We propose to ACRF to build a network of multiple of these devices that we could see the skin at quite detailed. You can even see the hair follicles here. So quite good detail, and we can follow patients with their skin in different time points. So we presented this consortium. We wanted 15 of these devices, 5 in Queensland, 5 in New South Wales, 5 in Victoria. We know that this is not enough to cover the whole population, but we wanted to demonstrate that this could work. We got a lot of partners in health in Queensland, New South Wales, and Victoria, additional partners that will help us in this. We got from the universities the commitment to over $2 million and in-kind support for example, my salary that I dedicate to this and salaries of many other researchers and people, we were offering $6 million. ACRF said that it was the best grant ever and gave us $10 million to purchase all these devices, connect all of them in a network, put them in the public system, get it into the e-health of each of the departments, and start building this database, this huge database, what we call the Imagenome. And the system now can, with some artificial intelligence, recognize moles and could create a map. So this is happening now. But there's a problem. I don't know if you can write the left, I can, but the right says that is the description that is in the ring, in the lot of the rings that say that there's one ring to rule them all. Well, that's what everybody wants. One single thing, not a magic bullet that could destroy a disease, anything that could solve all the problems, climate change. Well, that's not true. And not in humans. This is across course in humans. We have 30 trillion cells. And each of the cells, they have DNA, has RNA, and they have finally proteins. So we have between 20 and 25,000 genes that we know, 2 copies of each of these, and we have 10 billion proteins in each of our cells. We're really complex as an organism, and that's a single cell. So when we study genes, we talk about the genome as the object, and the discipline is genomics. If we've got a proteins, will be proteome, and will be proteomics. If it's images, will be imagenome, and it will be imagenomics. So what we want to do is that because in biology, there's not one omics to rule them all, we want to get multiple omics to give us all the information that we need to review our patients and identify on our patients. So we're gonna collect clinical data, different things about our patients. We're gonna look at genomics. These are patients that they have skin cancer. The patient on the left has some changes in the genome and means that the patient has 87 nevi and 3 melanomas. And you've got a patient on the right that has almost no nevi, only 1, and still has 2 melanomas because of a different genetic background. We're going to look at the histology of every single lesion, and we could do more tests on those histologies on the lesions that need to be removed. And my personal project, we are going to get scales from the surface. We're going to put a sticker, get the proteins that are on the top. We're going to analyze those and try to see if we can differentiate. We have some preliminary data, melanoma for just a normal nevi. So all these things we're going to combine. We want to get 15,000 people of low, medium, high, and very high risk of melanoma, all ethnicities. We want to collect over 60,000 avatars, more than 1.5 million lesions, although we say 1.4 in the grant, and create the world unique, largest, and most comprehensive skin multi-omics database. That's ACRF ASMIT. After all this data is collected, of course, we've got already our connection with people with big data and artificial intelligence. We're going to connect all this, machine learning, everything. This is all the people. I'm one of the 3 leaders, together with Prof. Peter Sawyer in Queensland and Victoria Mar in Victoria, in Monash University, and the rest of the partially part of the team. And this is what we want. At the end, you can go shopping, you step in one of these devices, you get one photo, then you walk, and while you're walking, you receive an SMS, and it's telling you your skin is okay, you're good to go, you have something, you need to go and see a dermatologist because there's something in some place. So when you see the problematic lesion and you don't understand what this lesion is, I can tell you, this is all good, there's no problem, okay? Thank you.
I also commend you on your efficiency. We have time for 1 to 2 questions.
Hi, Pablo, wonderful talk. I was looking at the numbers that you put there for the machines, and just making some numbers myself. You were talking about 15 units of that equipment. So I imagine because they're the first one of the kind, the price tag will be a bit astronomy based on that area. But you see how that could actually reduce in price, because at the end they're cameras. Cameras are cheap systems per se. So you see how that could reduce in price in the next 5 years, 10 years or so, with the number of demands, basically.
For sure. Each of the devices are around $500,000 Australian dollars. So that's the reason we need that big grant. And then we are going to invest a lot of money. We're investing already a lot of money in connecting all of them to the EMR, the electronic medical records, to a storage, what is called the RIS-PAC systems in the hospitals. And then all the information has to go to database centers in the 3 universities, and then we need to link all this. So there's a lot of costing for the research, but for the use, if someone else wants to buy one for home and play with that and see how this works. Okay, it's true. The device that we have now is 92 cameras with special flashes. The most important thing on imaging is that color should be constant. So we need that space, and then closest space at this stage is important. But undeniable, in the future, you get your phone, you start taking photos of the whole thing. You could develop some corrections of those images and get something similar. And these are DSLR, and DSLR, we know that the chips and the receptors of light, they're changing and dropping price all the time. So for sure, it's going to be in 5 years, the technology will be different. But everything that is behind to build this AI is what we want to do with the project.
Thank you. Pablo, we have another question online.
Thank you. Maria Victoria Rosso is asking through the chat, Vectra is being used only for research, or are they available in private practices or in public hospitals in Australia?
So the project, the ISMIT project at this stage is happening both in some private facilities and public facilities. It's still a research project for this, but certainly, for example, in our high-risk melanoma clinic, that we're following patients now with 2D photography, we're going to put all of them in 3D photography. So clinics that they have this device may use for their clinical assessment as a personal particular thing in the facility, but what is the research still is our research activity and is not approved for the use or diagnosis of patients. But yes, they could find devices because the project, the company has sold around 15 to 20 in the whole world, and we're buying 15 in Australia, so they're very happy with us.
Thank you, Pablo. Let's just put our hands together. Pablo.